What is up, you guys? It's another episode of Controversial Thoughts. As you all know, these are going on Instagram, they're going on YouTube, and they're also being released on my podcast every Friday. I've got so many controversial thoughts that I release them on Fridays on the podcast. We are super stoked at Heart and Soil about fire starter and gut indigestion, and I've been thinking a lot about lectins. And through Heart and Soil, I have gotten tons of emails asking about blood type diets. Is there anything to blood type diets? So I thought I would just do a whole video about blood type diets and debunk this once and for all. If you guys want the too long, didn't read, we'll talk about it all in this video, in this mini podcast on Controversial Thoughts. But it's going to wrap into a discussion of lectins and gut health. And those are very interesting to me. Those are things I've talked about before in my book. And I think they're fascinating in so many realms. So before we get into actually talking about blood type diets, let's just talk about blood types, because this is a really confusing thing that a lot of us didn't even learn that in that much detail in medical school. So there are many glycoproteins on the surface of our red blood cells that determine blood types. The major one is called the ABO blood type system, which is determined by a gene on chromosome 9, which is called ABO glycosyl transferase. And you'll understand why that's important in a moment when I show you the different sugar groups. It's a glycoprotein on the surface of our red blood cells. And type A blood, which is actually two different types of blood, type B, type O, which has three different types of blood, and AB blood are all different types of glycoproteins. They're all different sugar molecules attached to a protein on the surface of our red blood cells. So there's a base protein and there's different sugar molecules attached to that by this ABO glycosyl transferase on chromosome 9. You inherit ABO blood group genes from both mom and dad. They're codominant, and that gets a little interesting as well. But I'll review all that. I just want to let you guys know there are many other blood groups beyond that. There are 33 recognized blood group systems. They include things like Lutheran, Kell, Rhesus, Lewis, Duffy, Kidd, P, XG. There are so many different blood group systems out there. ABO is really just the tip of the iceberg, as you will, if you will, in terms of these. So anytime anyone tries to say that the foods you eat can be determined by something as simply as an ABO blood group antigen, you should ask them, what about the other 32 different types of glycoproteins on the surface of every human's red blood cell? It's way more complex than that. Basically, the take home here is that lectins are bad for everybody. That lectins from beans and grains and nuts and seeds and some other foods, some people eggs, some people dairy, can be problematic for most of us, mostly by interacting with our gut. But some of us, they do end up in our bloodstream, and they're not going to be uniquely bad for one blood type or another. That's way too simplified. I think that the benefits of a blood type diet are that most of the blood type diets eliminate the most harmful lectins in the first place. And if you eliminate things like grains and nuts and seeds and beans, you're going to feel better no matter what your blood type is. That's the take home, but we'll break it all down. I will break it all down for you in this here video. So I want to show you guys a little more about what makes a blood type and what it looks like. Again, if you're listening on my podcast, thanks for listening. It's on Instagram. It's on YouTube if you want to see the video of all this stuff I'm going to talk about. This is the ABO gene, the glycosyl transferase gene, the ABO glycosyl transferase gene. And this is what the different sugars look like on the surface of different blood types. And this is, again, simplified. So A, B, and O, and AB, but it's a little more complex than this, as I will show you in a moment. At this basic level, it's not even that simple to begin with. You can see that O has these four sugars. The diagram down here indicates those four sugars as galactose, N-acetylglucosamine, and an N-acetylgalactosamine. Now, if you're familiar with the nomenclature here, N-acetylglucosamine is just a glucose molecule with an acetyl group. N-acetylgalactosamine is a galactose molecule with an acetyl group on it. So glucose and galactose are very similar molecules. They're both hexagonal structures, sugars, and they're different just on the orientation of a hydroxyl and a hydrogen in those two. And fucose is a little bit different looking sugar molecule, but you can see that type O has these four sugars. Type B has an extra galactose on the end, and type A has an extra N-acetylglucosamine on the end. AB has both A and B glycoproteins on the surface. And this is important because there are antigens, or excuse me, there are really antibodies made to these in the human body. So a type B blood type is going to have an anti-A antibody. And that's important. A type A person is going to have anti-B antibodies. And type AB is not going to have either. And type O is going to have antibodies to both type A and type B blood, which affects our immunologic response to pathogens. And it affects which type of bloods we can get in transfusions, because if you give somebody blood that has a type of blood in it that you have an antibody to, it's going to cause problems. It's going to cause agglutination. It's going to cause hemolysis. No bueno at all. So why does this matter? Well, according to the ABO blood type diet theory, people with type A blood, which have this one different sugar molecule on the surface of their red blood cells, should eat different foods than people with type O or type B or type AB. To me, like I said, it's just way too simplified. There's no science to support it. In fact, there are studies to suggest that it doesn't really work. And evolutionarily, it doesn't make a whole lot of sense either. So one of the things I found interesting as I was researching all this stuff was that the original blood type for humans is not O, it's A. And it's just where we came from. So bonobos are exclusively blood type A by monoclonal antibody technology, and chimpanzees are blood type A as well. Now, that's just to say that our primate ancestors all have blood type A. Our primate ancestors all have a glycoprotein on the surface of their red blood cells that looks like this one. So this is the one I showed you guys earlier. It has this N-acetylgalactosamine at the end of the glycoprotein added to the protein by ABO glycosyltransferase, right? Now, what was the next one? I don't think anybody fully knows, but it looks like O was the next one to arrive about 2.5 million years ago. And then B and AB is apparently a very recent addition to the blood group antigen grouping. Some estimate that type AB has only been around for 1,000 to 2,000 years. So this is all quite interesting in the context mostly of infectious etiology. I think that one of the reasons the different blood types evolved was infectious agents. So let's put foods aside. Bacteria, viruses use these glycoproteins on the surface of our cells to attach. And there are certain blood types which are more susceptible or less susceptible to certain infections. So I think that it's more likely that blood types evolved in response to infections, and people with different antibodies to their blood types had more or less immunologic tolerance, immunologic ability to defend themselves against other infections. I should also note, we're getting a little complicated, a little technical. It's only a few minutes into this podcast, but you know how I like to roll. That there's another gene called FUT2, which tells you whether you are a secretor or a non-secretor. So you can secrete molecules of this ABO blood group locus into fluids of your body, into your intestines, into your saliva, into your semen or other fluids. This is actually used in police departments if there is a rape or a sexual assault and the perpetrator is a secretor. You can see what blood type they are based on their semen. We can see it based on saliva in people who are secretors. So it just means that those who are secretors actually secrete these glycoproteins into our fluids. And that probably gives those of us who have certain FUT2 genes to make us secretors a little bit more immunologic ability to prevent or protect ourselves from infections. Now, that's all well and good. How did anybody decide that this one was going to create one diet or another? And the fact of the matter is, it's pretty much just my made up. I mean, I appreciate Peter D'Adamo. He's a naturopathic doctor. I'm sure he's well-intentioned and very intelligent, but there's really no science to support which people with any blood group should eat what foods. Now, the reason this is such an appealing concept is because I think as humans, we like to think there's something individually that can unlock our destiny, that we can unlock the perfect diet for you or me based on some identity, but this is much too simplified. The reason it's such an interesting concept, and I think the reason it's become so pervasive, even though I think it's misleading, is because there is just a kernel of truth here. And the kernel of truth is that plants do contain lectins. Now, what are lectins? Lectins are carbohydrate binding proteins. I spoke about this in my book, The Carnivore Code. I'm going to review a lot of those articles here with you guys. But I'm going to start with an analysis of the actual science of ABO blood type diets, which won't take long because there's not much of it at all. So this is really the only review paper looking at blood type diets. The title is Blood Type Diets Lack Supporting Evidence, a Systematic Review. They did a large review of PubMed databases, and they found 16 articles, and that included blood type diets, but none of them actually met the study criteria. And they said no evidence currently exists to validate the purported health benefits of blood type diets. I would agree with this, but if you go further, this is a study from the American Journal of Clinical Nutrition in 2013. You can see all of the studies that they actually looked at, and I've looked at all these studies. Most of them are narrative reviews. There are 3 to 4 experimental studies here. The first one in 1997 was humans grouped according to the MNS blood group system. They gave them a low-fat diet, and the outcome was quantitative measurements of the reduction in LDL cholesterol. It doesn't really mean anything because it didn't really seem to support the hypothesis that it was helpful. So they said the identified article studied the variation between LDL cholesterol responses of different MNS blood types to a low-fat diet. However, the study did not directly answer the current question, which is whether ABO blood types and diet have any effect on health. So there was another experimental study in 1966. Humans were grouped by ABO blood group system, and they looked at serum concentrations of intestinal alkaline phosphatase. Didn't really offer a whole lot to us. 2012, humans were grouped according to ABO blood group system. There was no intervention, but we did see in that study, which was interesting, that microbiota profiles were different, which makes sense because 80% of people have a FUT2 gene, which makes them a secretor, which means that you will secrete your ABO blood group antigens into the gut, and that will affect your gut flora. So if you have a different, blood type, that could affect your microbiome. Does that mean that's going to affect the type of foods you should eat? I would say no. It's going to affect your microbiome, and that's something we should think about in microbiome research, and it will be wrapped into discussions later about lectins, but it doesn't tell us what foods you should eat. The other one is a 2007 study. Humans were grouped according to ABO blood group system, and they did food allergy tests with ELISA, MRAST, and LRA tests, and they looked for IgG, IgE, and T cell responses, but their results actually suggested something different than the traditional blood type diet that D'Adamo would suggest. So if you look at this blood types diet study, they got completely different results in terms of the IgE responses. So type A1, remember there are 2 different types of A, even more complex than just A, reacted most strongly to nuts and beans, eggs, dairy, and nightshades. Type A2, which D'Adamo doesn't distinguish, reacted the same but more strongly to dairy, eggs, and gluten grains. Let's just pause right there. As you'll see in this paper, everyone is reacting to foods that contain the most common lectins, that are things that I've talked about in the past, that are things that we should probably exclude on an autoimmune carnivore diet if you have an autoimmune illness. These are not unique foods to one blood type or another. These are the foods that I believe most people should exclude before including in their diet if you have an autoimmune disease. Nightshades, eggs, dairy, beans, nuts. I've been pretty clear in my messaging. I hope I've been clear that I think a lot of people with autoimmunity should do an autoimmune carnivore diet. What is an autoimmune carnivore diet? An autoimmune carnivore diet is grass-fed beef, organs, nose to tail. If you can't do that, get desiccated organs, hardened soil. We can help you out with that. And then bone broth, salt, and maybe one type of carbohydrate that's low lectin. You can use honey or something else if you want to. That's an autoimmune carnivore diet, in my opinion. It's much better than beef, salt, and water because it's more nutritionally replete, but it still eliminates everything that could cause immunologic reactions. When I'm working with people on a carnivore diet who haven't gotten better and they're still eating dairy or eggs, you got to get rid of those. They do have lectins. They trigger less people than nuts and seeds and beans and nightshades, but they can still trigger people. These are foods we would not have eaten a lot of evolutionarily. Some people can handle them, but a lot of people can't. And there's no evidence that there's any correlation between who can handle them and blood type. I think most humans cannot handle them regardless of blood type. And you'll see that in this study. Type B reacted mostly to eggs, nuts, beans, dairy, gluten, greens, nightshades, and sugars. That's like all the same as type A1 and A2. It's exactly the same. Type O reacted most to dairy, eggs, gluten, grains, and nightshades. That's almost exactly the same. Type AB reacted most to nuts, beans, seafood. That's a new one. Eggs, and dairy. Almost exactly the same. A2B reacted to gluten, grains. Now, what's interesting is this is, again, MRAST, IgE, food allergy testing, MRAST, IgG, T-cell food hypersensitivity tests, which are ELISA, ACT, LRA. And what they found was that across the board, people reacted to high lectin containing foods that probably should be left out of any diet that hopes to improve autoimmunity, at least in the beginning. You should do an autoimmune carnivore, an autoimmune animal-based diet if you have autoimmunity. It's why something like beef, salt, and water works well, but that diet, in my opinion, is just bereft of nutrients. You're going to get nutrient deficient with that diet. Eat nose to tail and do it in an autoimmune fashion. If you can't get the organs, get the desiccated organs however you can, or get them fresh from places like Belcampo, White Oak. I love those guys. Now, how many lectins are in our diet? They're common. They're very common. Lectins in the United States, a survey of lectins in commonly consumed foods, a review of the literature. Based on this survey, a review of the literature, we conclude that dietary exposure to plant lectins is widespread. This is a 1980 study in the American Journal of Clinical Nutrition. They're common. Where are they mostly? They're in edible parts of plants, dairy, and eggs. So if you really want to do a low lectin diet, you should probably cut eggs out of your diet. You should probably cut dairy out of your diet. Do you need to do it forever? No. If you get better, re-include it, see if you get triggered. I know personally with my eczema that if I eat any type of milk, whether it's raw, cow's dairy, A1, A2, goat's milk dairy, whatever, sheep, yak, camel, I haven't gone to the ends of the earth, but you get the idea. My eczema gets triggered. It's my immunologic phenotype that I will react to this dairy lectin no matter where it's coming from. Does that my blood type matter? No, I'm a B positive. The positive is a Rho. That's the rhesus antigen, which is something we're not going to go into in this mini podcast, but you get the idea. People react to lectins, period. Where are lectins? Grains, nuts, seeds, beans, dairy, eggs, nightshade vegetables are the biggest ones. That looks a lot like the plant toxicity spectrum that's in this book called The Carnivore Code that I wrote. You guys can check that out if you want. But those are the foods, aside from dairy and eggs, of course, that I think are the highest toxicity plant foods. Eliminate those, eliminate dairy and eggs, at least temporarily, if you're reacting to something, and see how you do. Then include the animal foods if you want. I think you're better off eliminating those plant foods forever. But the takeaway is that those are the biggest contributors to lectins across all blood types. And there's no evidence. There's not a shred of evidence that you can use ABO blood group antigens, one of 33 different types of blood typing, to determine who gets what. It works because for all of the blood types, D'Adamo recommends avoiding the most commonly ingested lectin high foods like beans, etc. If you don't believe that lectins are a thing, they're a thing. I'll show you a bunch of data to suggest this is a real thing, but they're a real thing. There's a lot of data to corroborate this. Characteristics and consequences of interactions of lectins with the intestinal mucosa. That's foreshadowing of a future paper I'm going to show you in this little mini podcast. Dietary lectins are metabolic signals for the gut and modulate immune and hormone functions. Not a good thing either. For a lot of people, that's a bad deal. Now, we're going to get all into that. I'm going to talk about Parkinson's. There's a lot more on this podcast, but just take a breath and think about everything that I have suggested so far. And I want you to consider red kidney beans and red kidney bean poisoning. So a lot of the studies I'm about to show you deal with red kidney beans. They deal with other lectin-containing foods, things like gluten, of which gliadin is a fragment, and gluteinins. Those are lectins as well. Concanavalin A, which is from lentils, and elderberry, Sambucus nigra agglutinin. So those are all, there's also wheat germ agglutinin, there's peanut lectins. What are all these? These are mostly plants, they're mostly seed lectins. Pretty interesting paper here that I'll show you. Lectins can induce in vitro release of interleukin-4 and interleukin-13 from human basophils. Dietary lectins present in beans and other edible plant parts pose a threat to consumers due to their capacity to induce histamine release from basophils. Histamine, our old friend histamine. In the next couple of weeks, we've got histamine and immune coming out from Heart and Soil too. So I'll be talking about this kind of stuff more in the future. But you can see here, concanavalin A, lectin, phytohemagglutinin from red kidney beans, and Sambucus nigra agglutinin, that is elderberry. And you see here that they triggered human basophils, immune cells, to release inflammatory cytokines, interleukins. They can enter circulation after the oral phase. Now, this is going to happen in everyone. Basophils don't have ABO blood group antigens in the same way that red blood cells do. There's no difference in blood type in your basophils. No matter who you are, these lectins from lentils, from red kidney beans, from elderberry are likely going to be triggering histamine release, interleukin release, triggering inflammation in all humans, Now, there's a really funny story about elderberry that I must share with you guys before we move on, because it's just too good not to. Elderberry is something that is widely promoted in the realm of coronavirus. I don't think it's a good thing for humans, but this is elderberry, and it's actually it's a deciduous shrub or small tree with berries. It's slightly toxic. It's slightly toxic when it's raw, and like many plants are, and you must cook it to make it less toxic. What's interesting is that there's an amazing story of this svelte gentleman. I'm joking. I'm showing a picture of Michael Greger, who is a pretty bloated, distended belly vegan physician who will not respond to requests for debate, who actually mentioned that he poisoned himself once with raw elderberry. And little did Dr. Greger know that raw elderberry has lectins, which are toxic, and he was drooling. It also has hydrocyanic acid, cyanogenic glycosides. So isn't that interesting and kind of ironic that the leader of the vegan world, who doesn't look super healthy, forgot that this elderberry was mildly poisonous. Cyanogenic glycosides. This is plants coming after you guys. You don't want these things, and you definitely do not want to look like Michael Greger. So don't eat a vegan diet if you value your body composition, your six-pack, maybe in general. Also, don't eat raw elderberries. So the other types of lectins that are commonly studied are things like phytohemagglutinin, which is the one from red kidney beans. And there are actually some pretty striking studies with phytohemagglutinin. If you've read my book, you know that there are many examples of red kidney bean poisoning. If red kidney beans are eaten raw, you will get super sick because phytohemagglutinin, this lectin in there. If they are undercooked, there are many incidences of people getting massively sick when they tried to eat undercooked red kidney beans. Beans, lima beans, whatever you want to eat them, whatever you want to call them, whatever type of beans, they're frankly toxic when they're raw. Plants do not want these things to get eaten. Now, if you go even further and you look at what some of these lectins are doing in the gut, it's pretty intriguing. Bacteria-lectin interactions in phytohemagglutinin-induced bacterial overgrowth of the small intestine. So now we're trying to connect lectins with GI issues, and so many of the questions I get now are about SIBO, small intestinal bacterial overgrowth. You can listen to the previous podcasts I've done with Lucy Mailing and others on this. There's really, I don't think there's any such thing as SIBO. It's mostly dysbiosis. It's an overgrowth of the wrong type of bacteria. It's a loss of normal diversity. And look, here is bacteria-lectin interactants in a PHA, phytohemagglutinin-induced dysbiosis. Is it possible that most dysbiosis we see in humans is caused by lectins? I think it's totally possible, and it's a very reasonable, strong hypothesis. Now, what's interesting is if they give phytohemagglutinin to rats and mice, those rats and mice do not thrive. They lose weight. They do not do well at all. And the mechanism of phytohemagglutinin-induced bacterial overgrowth of the small bowel of the rat was studied. What's interesting is they found that there was no specific association between the lectin and the bacteria. So why is, if that's true, why is phytohemagglutinin causing dysbiosis in this model? And I think there are other papers, again, all of this is talked about in my book, The Carnivore Code, which tell us why. So it's actually not an interaction with the bacteria. It's an interaction with the intestine directly. This phytohemagglutinin, these lectins, are directly toxic to our intestines. They show here, phytohemagglutinin feeding has been also observed to affect small intestinal growth in a negative way. These effects are believed to result from changes in the microflora induced by the presence of PHA in the diet, as germ-free animals do not display the same changes in conventional animals. They impaired growth. So here's what I said earlier. Causes reduced growth rates in several animal species when incorporated at 0.5 to 5% of dietary feeding. Lectin feeding results in diarrhea, impaired nutrient absorption, growth rate inhibition, can even lead to eventual death of PHA-fed animals. So what this paper actually starts to suggest is that the PHA is able to bind directly. So phytohemagglutinin, this lectin from beans and other lectins probably as well, lentil lectins, probably lectins from elderberry, are able to bind directly to the mucosal surface of the intestine at the brush border membranes. And that is probably the problem. I think that what's going on here, and there are multiple studies to suggest this now, is that lectins are binding directly to our gut, directly to the brush border, directly to the small intestinal villi, and probably affecting the production of mucin by the goblet cells. We know that the mucus layer of the gut becomes amorphous, it becomes fenestrated. There are holes that develop in the mucus layer. The flora from the gut, primarily gram-negative organisms like E. coli, then can touch the lining of our gut, and they proliferate and they cause inflammation, and thus you get dysbiosis, you get overgrowth of the wrong type of bacteria, mainly gram-negative organisms like E. coli, probably because the lectins are negatively affecting the brush border membrane. So this is the brush border of our gut. This is the single cell layer. This is the lamina propria. I've talked about this below the epithelium of our gut where immune cells hang out. And here's a blown up view. Here's a villus, like a little finger-like projection. Here's a crypt. Here's the lumen, the space between the villi. And here in the crypt are where the goblet cells live. And I think there's a pretty darn good hypothesis to be made, or a model system to be advanced here, that in the crypts, the lectins are negatively affecting the production of mucus by the goblet cells, and that is leading to dysbiosis. You don't want to eat these lectins. These are toxic food for humans. If you're going to eat them, you have to detoxify the heck out of them. You don't want this. You want a healthy gut lining. You want a healthy gut lining. You do not want leaky gut. You do not want these lectins, regardless of your blood type. This is lectins interacting with your gut. There is no blood type antigen here. If you're a secretor, then you are secreting the blood type antigens into your gut, but there are no red blood cells here to agglutinate. There's nothing. And these lectins are toxic, I believe, to all people, not just those who have certain blood types. And again, Diadamo eliminates the most toxic lectin-containing foods for people on all of the diets. So why would you include lectin-containing foods? I think most people are going to thrive with an animal-based diet, end of story. And that is where you get the major benefits from these type of ideas, these type of diets. Small intestinal growth caused by feeding kidney bean phytohemagglutinin lectin to rats. This is suggesting that in the small intestine, you're getting growth that could lead potentially to the formation of tumors. Like, we don't even know, but these lectins are affecting our gut in some pretty negative ways. If you have any doubts about these lectins showing up in the blood, there are multiple studies like this one, identification of intact peanut lectin in peripheral venous blood, that show that in humans, peanut lectin shows up in your blood. This is probably a bad thing for everyone, no matter what your blood type is. Again, remember I said in the beginning, the problem with blood type diets, the reason these are so confusing is because there's just enough truth here to confuse people, but there's never been a single study to show that one blood type is better than another with regard to these lectins. I think they're harmful for everyone, and everyone is going to benefit from an animal-based diet with low lectin foods. If you're sensitive, if you're autoimmune, eliminate dairy and eggs, do an autoimmune carnivore diet with one source of carbohydrates. If you want, it's up to you. I'm going to wrap up this little mini podcast with a real small soliloquy about Parkinsonism. This is probably the most fascinating part of this. So there are a couple of researchers, Brack and Hawk. This is one of the papers from Brack. 100 Years of Lewy Pathology, where he talks about alpha-synucleinopathies. Alpha-synuclein is a protein that is found in Lewy bodies. Lewy bodies are known to be associated with Parkinson's disease. Parkinson's disease in humans looks like masked faces, problems with movements, tremors. You guys have all seen Parkinson's disease, I'm sure. But Brack and Hawk had a hypothesis that there was something in our gut that might have been triggering Parkinson's disease. And there are a number of studies that corroborate this hypothesis. One of the most interesting ones was actually done in humans. And again, this is an associational observation trial. I talked about all this in my book, but what did they find? They found that in a Danish cohort who underwent vagotomy, this is severing of the vagus nerve, which goes from the stomach and the intestines to the brain, in between 1977 and 1995, they were matched with the general population, and they found that the risk of Parkinson's disease in patients who underwent truncal vagotomy, that is severing of the entire vagal nerve, the vagal nerve that went to both the stomach and the intestines, and then up to the brain, compared to those with superselective vagotomy. Superselective vagotomy is when they sever the vagus nerve that goes just to the stomach. And what they found was that those who underwent truncovagotomy, which is the branches of the vagus nerve from the brain to the intestines and the brain to the stomach, had a lower incidence of Parkinson's disease, a significantly lower incidence of Parkinson's disease. Again, this is just associational, but this is a very interesting finding. Full truncal vagotomy is associated with a decreased risk of subsequent Parkinson's disease, suggesting that the vagal nerve may be critically involved in the pathogenesis of PD, or Parkinson's disease. What are they suggesting here? Well, they're suggesting that, is there a substance in the gut that is actually able to move, to be transported, to transfer from the gut to the brain and be involved in the formation of Lewy bodies with these alpha-synuclein pathological formations of this protein? That would be amazing. What type of a compound could that be? I don't know. Maybe lectins? Is there evidence in other species to further corroborate this? There is. This is amazing, you guys. Ingestion of subthreshold doses of environmental toxins induces ascending Parkinsonism in the rat. What did they do? They gave these rats a compound to induce leaky gut. And they also gave the rats a lectin. They gave it a lectin, they gave it phytohemagglutinin, I believe, a number of other lectins, and they gave it paraquat to make the gut leaky. And they found that misfolded alpha-synuclein was found in different parts of the brain. Specifically in Parkinson's, we're worried about the substantia nigra and other neurons, but they found that vagotomy prevented the development of Parkinsonian symptoms and constrained the appearance of misfolded alpha-synuclein to myenteric neurons. What they're saying here is that when they gave these rats leaky gut by giving them paraquat, and they introduced a lectin, a number of lectins from things like beans or concanavalin A, they found this actual lectin in the brain with the presence of misfolded alpha-synuclein and neuronal loss in the substantia nigra and the dorsal motor nucleus of the vagus nerve. How crazy is that? And then they could prevent that. They could prevent that with vagotomy, just like they saw in the Danish cohort. Again, the Danish is just a cohort. It's environmental, observational data. It's not an actual experiment, but wouldn't that be amazing if lectins from our gut were traveling through the vagus nerve like they do in this rat study? There's another study in C. elegans that shows the exact same thing, and you can see the lectins in the brain of these animals with the dopaminergic neurons of the substantia nigra, and these animals exhibit symptoms of Parkinson's disease when they are given these toxins. Interestingly, in that first study I showed you with rats, when they just give paraquat, the animals don't get Parkinsonian symptoms. It's only when they give paraquat plus lectins, plus these things found in beans that the majority of the world is telling you to eat because they're healthy. They're not healthy for anyone, regardless of your blood type. They're not healthy for anyone. Like I said, there's a study in C. elegans, these worms, that show the exact same thing. You can see the lectins moving through the vagus nerve to the brain. When they cut the vagus nerve, they don't end up there. So there's a really strong set of data here. The Danish cohort, Brack and Hawk's hypothesis that something is moving from the gut to the brain to induce this formation of Lewy bodies in Parkinson's. Is it lectins? That would be crazy. You in your life probably know someone that's affected by Parkinson's. My grandmother had Parkinson's. This is one of those diseases that hits home for me. Isn't it crazy to think that things in our diet, wheat, nuts, seeds, grains, legumes, things that don't want to get eaten, the most toxic parts of plants, could be contributing to lectins that are going to move through our gut across the vagus nerve, regardless of your blood type, and trigger the formation of pathological alpha-synuclein formations, alpha-synuclein clusters, these Lewy bodies in the brain, leading to the death of dopaminergic neurons in the substantia nigra, the dorsal motor nucleus of the brain. This is crazy. This is so interesting. Now, there are other lectins that have been shown to do this too. Not necessarily in the same models, but other lectins have been shown to be negative for humans, like gluten. This is gliadins. This is in vitro, so it's in test tubes, but here's an in vitro cytotoxic effect of wheat gliadin-derived peptides on CACO2 intestinal cell line, and it's associated with intracellular oxidative imbalance. Implications for celiac disease, you get the idea. So gliadin had an anti-proliferative effect. It was associated with intracellular oxidative imbalance characterized by an increased presence of lipid peroxides augmented oxidized relative to reduced glutathione. I've talked about that a lot. And a loss in protein-bound self-hydryl groups. Not a good thing. Tell me more about why you want to eat wheat. Tell me more about why you want to eat bread. If that weren't enough, there is actually a large body of evidence to suggest an association between bread and mental illness, between bread and schizophrenia. There was a shortage of bread in Sweden, I believe in the 40s, and there was a decreased incidence of schizophrenia in that time. Again, it's correlation, but isn't that interesting? The use of a gluten-free diet in schizophrenia. Why are we using a gluten-free diet? Because gluten is gliadin and glutenin, lectins that damage the gut. Could they move through the vagus nerve like other lectins to affect the brain? I think you could. I think you could. It's really interesting to think about this. Now, I'm not saying that a gluten-free diet is going to cure schizophrenia, but this is a striking possibility. And again, what we have here is a lectin-associated pathology in the brain. One more, diet and psychosis, a review. People are thinking about this. Lectins are not good for humans, regardless of your blood type. Regardless of your blood type, we should be eliminating the highest lectin foods. I'll review them once again. Nuts, grains, seeds, legumes, nightshades. If you're still sensitive, eliminate dairy and eggs. Autoimmune carnivore. What is autoimmune carnivore? Grass-fed meat and organs. If you can't get organs, use desiccated organ supplements. We make them at Heart and Soil. We do the best we can for you guys. heartandsoil.co.co. We just released Firestarter and gut and digestion. I'll talk more about that one in a moment. Bone broth, get nose to tail, get methionine, get glycine to balance your methionine. Use salt. I like Redmond Real Salt. And use carbohydrates if you want. I think things like honey, I talked about in the controversial thoughts last week, are great. Or if you don't respond to fruit, fruit is going to have a lot less lectins than many other foods. That is my idea of an autoimmune carnivore diet, but you must eliminate the lectin-containing foods. None of this has anything to do with your blood type. Yes, you can be a secretor. You can have food too. You can move those antigens, those glycoproteins into the gut. But to say that we know what someone should eat based on blood type is based on zero science. There's actually science to say it doesn't really work. The biotype study found that everybody was susceptible to everything for the most part. And all the foods they were usually susceptible to are the foods I'm recommending that you guys eliminate. Regardless of your blood type, if you are not thriving, eliminate the highest lectin foods and you will thrive. You'll do better. Get the nose to tail, get organs, get them fresh, get them desiccated if you need. Why am I stoked about Firestarter? That's stearic acid. Balancing stearic acid and linoleic acid is hugely important. I've done many videos on that. Why am I stoked about gut and digestion? Because we just released this one too. It's tripe and intestines. So it's giving you actually the digestive tract, which contains the nutrients that you're going to need to make a healthy digestive tract. And the epithelial layer, all of those unique proteins, unique peptides. I've talked about this in the past. BPC 157 is found in the gastric juices. A lot of people are taking oral BPC 157, but I do think that a supplement containing intestines and tripe, our ancestors ate the intestines as well. They made sausages, they made stomachs out of this. These are valuable pieces of nose to tail as well. We combine it with liver, pancreas, and spleen. So you get all the digestive organs. The nutrients in those will also help fortify the gut lining. How do you fix leaky gut? You remove the things that are causing it. You remove lectins, regardless of your blood type. There's no data to support blood type diets. Remove the lectins, give your body the nutrients it needs with an autoimmune carnivore diet with things like liver, heart, kidney, spleen, pancreas, stomach, and tripe. We've got the gut and digestion supplement, which is out now. Check that one out. You can always email me, Dr. Paul, drpaul@heartandsoil.co if you have questions. Hopefully this is helpful. I think lectins are fascinating. They're freaking scary, but they're fascinating. And I think there's a lot of evidence that these are playing a role in human health and that we should be avoiding them. As much as possible. And the other thing to take away from this is autoimmune carnivore. If you are not thriving, eliminate eggs, eliminate dairy. These are not uniquely benign foods for humans. Some people can tolerate them, but not everybody can. So hopefully that's helpful, you guys. Stay radical. Again, this is going to go on IGTV. It's going to go on YouTube. It's going to go on my podcast after that. Heartandsoil.co, Dr. Paul at heartandsoil.co if you have questions. Love you all. Stay radical.